Hello, my name is Amanda Linskins, and I'm an undergraduate research assistant at the Doe Lab within the University of Oregon. My mentor is Kristen Lee, and my project aims to understand how temporal transcription factors influence neural circuit development in fruit fly larvae. Very little is known about the developmental mechanisms generating neural circuits. To investigate this, I focus on a well-characterized locomotor neural circuit, the Moonwalker Descending Neurons, also known as MDNs, and the Pair1 Neurons. As you can see in Figure 1, the 4 MDNs are located in the brain and control backwards crawling in the larvae and backwards walking in the adults. When these neurons turn on, they activate 2 Pair1 neurons, which control pausing in both larvae and adults by inhibiting forward locomotion. Like all other neurons, the MDNs and Pair1s are formed from neuroblasts, also known as neural stem cells in vertebrates. As neuroblasts develop, they divide into neural progeny and progress through various temporal windows marked by unique temporal transcription factors, also referred to as tTFs. The temporal transcription factors we will be focusing on include hunchback, crupple, castor, and grainy head, as seen in Figure 2. A neuron's identity and function is, in part, determined by the temporal window the neuron is born in. This leads us to my big objective. First, determining which tTFs the MDN and Pair1 neurons derive from, and second, examining how the MDN Pair1 circuitry is affected when the tTF is manipulated by assaying morphology and behavior. I will walk you through the methods and results for the first objective and then the second. My experiments rely heavily on the Gal4 UAS system, which can be seen in Figures 3, 4, and 5. When the neuron-specific enhancer is activated, it will allow the transcription of Gal4, as these proteins will then bind to the UAS binding site and activate the transcription of the linked transgene. To determine what temporal window MDN and Pair1 are born in, I used Gal4 UAS to express the green fluorescent protein, GFP, in MDN or Pair1 neurons individually, as seen in Figure 3. The tTFs were labeled with antibodies to observe overlap in the MDN and Pair1 neurons. The GFP labeling the MDNs is seen in Figure 6a, and the Pair1 neurons is seen in 7a. Figures 6b and 7b show the hunchback staining, and Figures 6c and 7c show the GFP hunchback colocalization. Both the MDN and Pair1 neurons overlap with the tTF hunchback. All tTF data was quantified in D, which supported that MDN and Pair1 neurons derived from the hunchback temporal window and not crupple, castor, or grainy head. Now that we know MDN and Pair1 neurons derive from the hunchback temporal window, I knocked down an overexpressed hunchback in the Pair1 neurons and assayed Pair1 morphology and behavior. As seen in Figure 4, in addition to expressing GFP, a second UAS binding site was added to generate the knockdown or overexpression of hunchback in Pair1 neurons specifically. Additionally, as seen in Figure 5, we utilized the optogenetically active cation channel to activate Pair1 during red light exposure. This was accomplished by treating the larvae with all trans retinol, also known as ATR. The behavior of the ATR group was compared to the behavior of control larvae treated with vehicle. In Figure 8, the Pair1 neurons are labeled with GFP in green, and in magenta is the NC82, which is labeling synapse-rich areas. In A is the control, where we can see normal morphology, 2 cell bodies, and the axons descending along the side of the neural pill. In B is the knockdown of hunchback, which resulted in axons failing to descend fully. In C is the overexpression of hunchback, which resulted in a fully developed third Pair1 neuron. These morphology changes can be translated into behavioral changes, as seen in Figure 9. The x-axis represents time in seconds, and the y-axis represents speed of the larvae. The blue line on the graph is the ATR group, which should experience pausing, and the white line is the vehicle group, which should not experience pausing during the red light, which is represented by the red box. As anticipated, in the control with normal hunchback, shown in A, the control vehicle group does not respond to red light, while the ATR group exhibits a pause. In B is the hunchback knockdown, where the ATR group pauses similar to the control, but remains at a slower speed after the light turns off. In C, the hunchback overexpression, which shows that while the ATR group pauses similar to the control, the speed immediately begins to increase rather than waiting for the light to turn off. With these results, I conclude that both the MDN and Pair1 neurons are derived from the same tTF, hunchback. Changing the amount of hunchback in Pair1 neurons affects morphology and behavior. These morphology and behavior phenotypes suggest that normal Pair1 circuit function is altered by changing molecular origins. I hope to further this research by determining morphological and behavioral changes with hunchback knockdown or overexpression in MDNs and adult Pair1s and MDNs. I also hope to perform neurite mapping of the Pair1s when hunchback is knocked down or overexpressed to see exactly how the neurite's morphology is affected. I'd like to thank everyone at the Doe Lab, SPUR, and the NIH for funding this research.